So congratulations on your CXC passes, guys. I know that the fact that you're here, interested in CAPE biology, means that you were successful in the CXC biology and you want to further your knowledge in biology. And really thank you for attending, because here at Extra Time, we have been around for the last 10 years, and our aim or our mission has been to empower students who really want to excel and master in the subjects of chemistry and biology for fourth, fifth, and sixth form. And it's not just about the fact that students want to pass the subject, but we are really designed to empower the students who want to excel and master the subject. And usually persons who want to master the subject are trying to enter into a competitive specialty, like for example, medicine or engineering, or you're trying to earn a scholarship. And so you want to have very good grades for that purpose. So we are here to encourage and empower students like that to aim for and achieve the distinction, and not only to achieve a distinction, but to also be listed amongst the students that performed, top students in the Jamaica and the top students in the Caribbean. And what we have found for the students that we have had on the merit list who have been found in the top 10 in the Caribbean and who have topped Jamaica, that really and truly to excel, you really need to go through 3 stages. There is a stage of preparation where before you actually start studying the content, you need to first review the curriculum of what it is that you're going to be preparing for. You need to have an overview of it. You need to understand how you're going to be graded, how are you going to be assessed, so that even before you start learning your first topic, you have an outline of the scope of work that you have to do in the time that you have left to prepare it. When you start off with an understanding like that, with a foresight like that, it tends to dictate your behavior. You'll find that you'll be more focused, you will know more of what is at stake, you will move at a more steady pace, because you are aware of the overall picture of what you're working towards. And you're not just waiting or depending on teachers to spoon feed you, hoping that the teachers are indeed giving you what you need to get without you ever checking, are you covering the content sufficiently? So this seminar is geared at giving you that information for your preparation phase, so that you understand the scope of Unit 1 chemistry, you understand how long it might take you to prepare, and therefore from the very get-go, you understand that you have to go at a steady pace. You cannot start slowly and then accelerate at the end. It doesn't give you that opportunity, whereas in CXC, sometimes persons may have been able to pull that off, where they did the bulk of their studies or the bulk of their work in the last week or so, or the last 2 weeks. CAPE is different. CAPE, because it is so complicated, and not, I wouldn't say complicated, but because it requires more advanced knowledge, you have to actually be comfortable and intimate with the knowledge from day one, because it can become overwhelming if you leave it to the end. So I'm Miss Smith. For persons who are meeting for the very first time, welcome, and let us get into it. So for the CAPE Unit 1 Biology, this is taken from the syllabus as to giving you an overview of what biology is from the CAPE perspective. So we know it is a study, it is a scientific study of living organism, and the study of this subject leads to an understanding and appreciation of the concepts of life at all levels, and hence to a greater respect and reverence for life. We understand the interconnected web of life and the unique role of the human species, that they are integral to the dynamic value of the biosphere. So the CAPE biology syllabus prepares students to acquire knowledge about how to protect, sustain, conserve, and improve the variety of life in the ecosphere. It is the foundation for persons wishing to pursue careers in biological, environmental, agricultural, medical, paramedical, and applied sciences. So this is the one foundation for branching off into these various sciences. And so the curriculum is designed to prepare you for these. So why do we study biology? All right, let me just play this clip for you.
We've got 60 seconds. Are you paying attention? Biology is the reason your body works and looks the way it does. The reason that your heart beats and your lungs breathe without you even thinking about it is because of your brain and nervous system. The reason your eyes and hair are the colour they are is because of your genetics. The reason you can eat an apple and turn that apple into enough energy to go for a walk is because of all the body systems, like your gut and your blood system, that can turn that matter into energy and that energy into movement. The reason that you age is partly biological. Whether you become 100 or not is down to your biology. The reason you walk, talk, are happy or sad, the reason you are you is because of biology.
All right. So it basically was giving you the perspective that as it concerns us as living organisms, all our habits or behaviors are determined by the biology, which is the structure and function of what we contain as living organisms. Now, as to the structure of your CAPE Unit 1 biology syllabus, unlike in CXC where your syllabus was arranged in sections, in CAPE, your syllabus is arranged in modules. So you have 3 modules underneath Unit 1 biology. Well, first, this module is concerned with the knowledge of biological molecules, reproduction, and development. So your module 1 consists of cell and molecular biology. Module 2 consists of genetics, variation, and natural selection. And module 3 consists of reproductive biology. Reproductive biology, usually everybody enjoys it, and it is one of the shorter modules because it only has 2 topics. But as you will learn in CAPE, right now in CAPE, it's not about having a lot of topics to study. They have significantly reduced the number of topics, basically less than half of what you would be studying in CXE. So no, it's not about width of knowledge, it's about depth of knowledge. It's about you being able to delve into a specific area, zero in on it, and perfect it. So that's what you're going to be doing in CAPE. You're going to be going deep. So now, at this level, when you're more advanced and more mature, you're now being exposed to depth of study. All right. So don't feel like, oh, it's easy because there are less topics. No, you're going to be asked more detail. Now, let us talk about module 1, which deals with cell and molecular biology. So this module has 4 major topics, and these major topics do have subtopics. So you're going to learn about aspects of biochemistry, and biochemistry basically deals with the properties of molecules which are essential to our survival. So that's us learning about the properties of proteins, the properties of fats, the properties of carbohydrates, and these are molecules. So in the first topic, you're not going to be learning. In the first topic, you're not going to be learning about an organism. You're learning about molecules that are found inside of the cell. And so there is a little bit of chemistry involved because, as it said, biochemistry. So a little bit of the chemistry knowledge is required here as we look at the structure of carbohydrates and how they react, and the structure of fats and the structure of phospholipids and the structure of proteins. So that's what we're going to cover in the first topic in module 1, aspects of biochemistry. Once we're done with that, the second topic would be cell structure and function. Here we're going to spend the time and we're going to learn about different types of cells, which we were introduced to in CXE. We learned that we had prokaryotic cells and eukaryotic cells. Well, now we're going to go deeper into that in CAPE Unit 1 biology, learning the features or the organelles in prokaryotic versus eukaryotic cells, how they differ and how they came about and all of that. We're also going to be learning about membrane structure and function. So whereas you in CXE, as I said, CAPE Unit 1 is all about depth of knowledge. So it's going to take something that you would have just glossed over it in CXE, and it's going to take it and it's going to go in detail. So in CXC, when you were learning about the parts of the cell, yeah, you did learn about the cell membrane, that it was a semi-permeable membrane. But in CAPE, you're going to learn the details of the cell membrane and realize it's way more complex than you thought it was. You're going to learn how it functions, etc. We're going to learn about enzymes again, building on what you learned in CXC, of course, going in significant detail. So module 1 is primarily based upon cell and molecular biology. Right? The molecules inside of the cell and the structure of the cell. So we have 4 main topics in module 1. Just letting you see the general objectives of this module is that, and going in deeper. So we want our students to learn the structure of water, carbohydrates, lipids, proteins, their roles in the environment. You have to understand the basic, that cells are the basic unit of life, relate the organelles of cells to their function, understand the fluid mosaic model of the cell membrane, and understand the mode of action and action of enzymes. So I have a video here to talk about the first topic in module 1, which is aspects of biochemistry, just to give you an application of this topic.
The shape of proteins. There are 4 different levels of protein structure or protein shape: primary, secondary, tertiary, or the 3rd level, and quaternary, or the 4th level. The primary or first level of protein structure is a chain-like or linear sequence of amino acids joined by peptide bonds, as you can see here. The secondary or second level has 2 shapes: an alpha helix or coil-like formation, or a folded pattern called a beta sheet. In both of these structures, hydrogen bonds between the amino acids stabilize the shape of the protein. The tertiary or 3rd level of protein structure involves more folding and bonding of the secondary structure. The coils may even touch each other as some covalent bonds form from the sharing of electrons between different amino acids. But most of these twists and folds occur from the result of ionic bonds between positively and negatively charged R groups of the amino acids. The quaternary or 4th level is where clusters of more than one polypeptide chain link together to form a giant molecule. Many different types of bonds may be formed within this structure. The shape of protein is important. For instance, fibrous proteins are extended linear proteins that are part of ligaments, tendons, and muscles. And globular proteins fold into almost spherical shapes and have their hydrophobic or water-fearing R groups buried deep within the core, and their hydrophilic or water-loving R groups extended out into the water when in an aqueous environment. Proteins take their shape based on the job they're required to perform, and some have moving parts that are important to their functions.
All right, guys. So the reason why we showed you that was because I wanted you to see the level of detail that you're going to have to go into. So yes, in TXC, you were told about proteins and you were told that they're made from amino acids. But no, you're going to be learning much more greater detail. You're going to learn the 4 types of structures of protein, and you're going to the molecular levels, because we need to understand the structure of protein so we can predict the behavior. We're also going to be going in detail in enzymes. And here's an application of why we're going to study enzymes in detail. We're going to learn how enzymes function, their structure. We're going to learn things that can slow down or prevent an enzyme from functioning, and some of the things that make enzymes work better. To some extent, what you learned in CXC, but in much greater detail. Here's the application of the topic of enzyme. One of the applications, that is.
Hey everyone, we're here with another video. We have all watched that movie scene. And you would be wondering, how did he die so fast? Bullets take longer to kill. Well, to understand that, you need to know the mechanism of cyanide poisoning. It's much simpler than you think. Human body is made up of cells. Except red blood cells, all cells have a part called mitochondria. Mitochondria is responsible for production of energy. Now, imagine a cell to be a car. The mitochondria will be its engine. Like an engine, it needs air or oxygen. But instead of petrol or diesel, its fuel is glucose. When cyanide enters into body, it inhibits the supply of oxygen to the cell by blocking its path, which chokes the cell and stops the energy production. As the cell is devoid of energy, it dies. The same thing occurs throughout the body really fast, killing the cells, and ultimately the person dies. Though the exact time of cyanide poisoning depends on a lot of factors like dosage and body weight. Different sources say it can take from several seconds to several minutes. Cyanide poisoning can also occur due to fire hazards and industrial exposure. In such cases, you need to take the person out in airy surroundings with a lot of fresh oxygen. You can also give the person sodium bicarbonate or baking soda. Though it is highly unlikely that you will ever encounter such situation, but who knows, you might even save a life with that knowledge.
All right, so this whole topic of cyanide poisoning is an application of the knowledge of enzymes. When you learn that cyanide is one of the inhibitors of a key respiratory enzyme, which is why you see it in the James Bond movies that they have cyanide tablets that if they get caught, they just take the cyanide tablet and die. But we will learn about more inhibitors also. Why we have a lot of chemical weapons like nerve gas and so on, is because it acts at the enzyme level. So we will learn all of this when we look at enzymes. So that's module one. So module one would cover learning about the biological molecules, learning about different types of cells, learning about enzymes, and also it covers another topic right here, which is membrane structure and function. So once we're done with that, we would move on to module two. So module two now, this module deals with genetics, variation, and natural selection. So we're going to spend some time learning about the two nucleic acids in the body, which would be DNA and RNA. So we're going to learn details about their structure, functions, roles. We're also going to learn how cells divide, the mechanism of how cell divides. We're going to look at meiosis and mitosis, and how it is that we end up having variation or variety in organisms that are in the same species. We're going to also look at patterns of inheritance. So patterns of inheritance. So we're going to look at how we pass on traits from one organism to the next. So we actually did this in CXC in genetics, but this one is going to be a dihybrid cross. Dihybrid. So it means that we're not going to only look at one trait alone. We're going to look at two traits being inherited at the same time. So that's going to be a little bit different from what you learned in CXC, because we're looking at two traits at the same time. And we're going to learn other more specific things to advance biology. Then there is also the genetic engineering. We're going to learn how it is that we can manipulate the DNA of organisms to bring about new features in that organism or new traits. So we learn how it's done, and even how we produce the protein insulin, hormone insulin. And then we look at natural selection, which is how a species becomes adapted to its environment. We look at Charles Darwin theory and see how what is the mechanism for natural selection. So for module two, the objective that CAPE wants us to achieve is that we should understand the structure of nucleic acids, which is the RNA and the DNA, and understand their roles in making proteins and its nuclear division. We need to understand the behavior of chromosomes, nucleus, and cytoplasm in mitotic and meiotic division, and their importance for stability and variation in a species. We need to understand the patterns of inheritance, understand selected aspects of genetic engineering and its medical, agricultural, environmental, ethical, and social implications. And we should understand the genetic basis for variation and its importance in natural selection. So I have a few videos to show you from things that are applicable for module two, starting off with this one, which comes underneath patterns of inheritance. We're going to see a pattern of inheritance, which we didn't cover in CXC, which is, this is called epistaxis, or epistasis, sorry. Let me show you this video.
Same things are occurring, but the way they are having higher structure or high length and all these things, right? Now what is epistasis? Now epistasis is a phenomena when one gene completely masks the effect of another gene, right? So it's just like one gene is playing the vital role and the master key role over any other gene. For example, if we look at here, in this case, coat color in mice is controlled by two separate genes, right? C and C. So capital C is for the pigment, which is pigment is present, or when the pigment is added, or small c is no pigment is present. Capital B for more pigment, small b for more pigment, or which is black, and small b is for the less pigment, which is brown. So you can see that capital B means black, small b means brown, but if capital C, that is the pigment is there, if small c, no pigment is there. So what it does actually, the small c here is acting as the epistatic genes. That means if there is no pigment present, even if you have this capital B gene in your character, in your gene, in your DNA, you shouldn't have expressed that, because there is no pigment present. Though you are having the gene color for black, but if there is no pigment, there will be no coloration. So that is what is called the new characteristic. If you are having small c, neither you will have a black coat because there is no pigment at all. It will give a complete different color characteristics, which is called albino or totally white. So if you look at here in this Punnett square, you can find that the production of albino characters, because due to the interaction between.
All right, guys. So this is it's a topic that you're going to learn when you get to module 2, when you're looking at patterns of inheritance. You're going to learn that it's possible that one gene can suppress another gene. That's known as epistasis or epistasis. And so when you are studying this type of trait, you have to look at the two different genes because one gene can suppress the other one from being expressed. So this one, when there is epistasis, it allows us to see something unusual because normally we're accustomed to seeing like black mice or we would see white mice, but then we are now able to see a third type of mice, which is black or brown, and we are seeing a third type, which is albino, because they have no pigment. But this is because of this phenomenon of epistasis, which you will get to in module 2. Then there's genetic engineering, which is how we are able to manipulate the DNA of organisms. We are able to insert new genes into people's DNA or organisms' DNA and allowing the organism to have a trait that it never had before. This is a very revolutionary technology because previously, if you wanted a trait to get into an organism, you had to do it by mating or breeding the organism with another organism from its own species. So if there's a trait that you wanted in a cow, like if you wanted cows to have a lot of milk, then you'd have to mate a bull and a cow. Hopefully they had a lot of, the cow has a lot of milk with a bull that has good traits and hope that the offspring will get the trait that you want. But genetic engineering bypasses that process of mating. You don't have to mate organisms together anymore to get a trait into an organism. And not only that, you don't even have to stay within the species. You might have a trait that you saw in the goat and you want that trait in a cow. And genetic engineering allows you to do that in the lab and manipulate the DNA of the cow and get the trait from the goat. So genetic engineering has revolutionized biology by breaking down the species barrier that we had before. Because in nature, a cow and a goat cannot mate, but genetic engineering bypasses all of that. Just a second here, guys. Let me just answer a question in the chat. So guys, what we find is that genetic engineering has given us another pathway that never existed in nature before, where we can take DNA from one species and insert it into a different species and cause the species to have a different trait than it ever had. So that is an entirely new discipline for us. So let's just look at applications of this knowledge.
This group in Edinburgh is part of the UK Cystic Fibrosis Gene Therapy Consortium, and for over 10 years now we've been working on developing gene therapy from a purely lab-based concept to something that we can actually use in the clinic to treat people with CF. So we're at the point now of having all the necessary funding and infrastructure to start the trial.
Cystic fibrosis affects all the cells in the body, but its effects are most felt in the lungs and in the gut in most patients. Cystic fibrosis affects children and adults with cystic fibrosis mostly by causing them to have a buildup of mucus within the lungs, and that buildup of mucus becomes infected, and once it becomes infected it begins to damage the lungs, and that damage ultimately leads in most people with cystic fibrosis to an early death because of respiratory failure. 130 patients are planned to take part in the trial, which will take place in clinical sites in London at the Royal Brompton Hospital, here at the Royal Hospital for Sick Children in Edinburgh and Western General Hospital.
Patients that are recruited to the trial are asked to come here to the Western General to a specially designed cubicles in which we give the nebulized treatment. And when they come for a treatment, we're giving this treatment by inhalation, so it's in a liquid form, and we put it in a nebulizer, which is this device here, powered by compressed air. It's taped up so they can't actually see the medication because it's a placebo-controlled trial and not be able to tell if they're on active treatment or dummy treatment. And they will inhale from this in the chamber specially designed for the purpose.
The idea is that when we nebulize this product into the lungs of patients, it then enters the cells of the patient's lungs where it is expressed as a protein, and this is the protein that's defective in people with CF. So now we have in their cells a working version of the protein that they have that's defective, and we anticipate that because of that their lung condition will improve, and we know that this is likely to happen because of experiments we've done obviously in the lab, in cells and so on in our preclinical work.
This production.
All right. So guys, essentially what it's showing you right here is that here's a disease called cystic fibrosis where the person produces very thick mucus and that plugs up the bronchioles and it causes them to get a lot of respiratory infections. And it's all because of a faulty gene they have. And here it is that they're using genetic engineering to deliver a good copy of the gene to the cells in the respiratory tract. I'm hoping that the cells will take up the good copy of the gene, use the good copy, and will produce the normal mucus, and the person will get relief. And to some extent, guys, it has been successful when persons have been treated. It's called gene therapy. They have been given a good copy of the gene, and there have been improvements. But we're going to learn in Module 2 what are some of the pros and cons of genetic engineering and the social and ethical implications of using this technology, which is foreign, because as I said, this is not nature's way of doing things. So this is definitely a synthetic man-made way. And then finally, guys, we have Module 3, which only has 2 topics, which is learning about how reproduction in plants occurs and how it occurs in humans. So again, it's only 2 topics, so it means that we're going to learn it extensively in detail. There's no detail that we're going to left that's going to be undone. So by the time we're finished with these 2 topics, you should know details about the reproduction in plants and in animals. So the objectives of Module 3, CXC wants you to understand sexual and asexual reproduction in plants and understand sexual reproduction in humans. So here is a snippet of some of the things to expect in Module 3 when we study reproduction in plants.
Double fertilization. S.G. Nauschen, 1898, was the first to show that both the sperms released by a pollen tube are involved in fertilization. They fertilize 2 different components of the embryo sac. The phenomenon is unique to angiosperms and is called double fertilization. The nucleus of one of the sperm fuses with the egg nucleus, thus completing syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the 2 polar nuclei located in the central cell and fuses with them to produce triploid endosperm nucleus. As this involves the fusion of 3 haploid nuclei, it is termed as triple fusion. Since 2 types of fusion, syngamy and triple fusion, takes place in an embryo sac, the phenomenon is termed double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell, PEC, develops into endosperm while the zygote develops into an embryo.
All right, guys. So this aspect of it, we're going to learn as we go into reproduction in our plants. We're going to learn double fertilization as we were seeing in this video. We're going to learn even more. We're going to learn how the plant produces pollen. We're going to learn how it produces the egg. We're going to learn the structure of the flower. So there's way more detail. As I said in CXC, you would have glossed over some of these things. Now you're going to go in detail. So that's reproduction in plants, and then there's reproduction in humans. This is an interesting aspect I found for this topic, which is looking at the possibility of making male birth control pills. But of course, if you understand the reproductive system of males and females, that is when you can try to manipulate it and prevent conception. So let's look at this one.
Oh, that means it's time to take my birth control. But it's just been making me gain weight and feel nauseous. Plus, I've had horrible mood swings, and I've been having the worst migraines lately. I just don't want to take it anymore.
My girlfriend takes her birth control every day, and I know she hates taking it. She complains about it to me all the time, and we do use condoms, but we want to be extra cautious. I feel like I should do something too, but I'm not sure what. Oh, I see condoms and a vasectomy, but we're already using condoms, and a vasectomy is just out of the question. Wait, here are some other options that look promising, but I feel like I should get an opinion from a professional.
All right, so this development of a male contraceptive pill is actually in development, whereby it would take off some of the pressure from the females, because right now, guys, most of the contraceptive methods are designed for females. Previously, the only thing males would have to do would be to use a condom or get a vasectomy. Females, because we're the ones who do have conception, or we are the ones who do have the pregnancy, there's a lot more methods available to us. But the development is there. A male contraceptive pill is in development. So guys, having looked at what each module entails, module 1, 2, and 3, we see that starting off from this week to the end of April, there are really approximately 34 weeks available, or call it 32 weeks available from now to the end of April. But there are at least 6 weeks out of the 32 weeks which you don't have school. So you have a December break, you have Heroes Week break, you have Easter break. And so really and truly in terms of teaching weeks, you really only have about 26 weeks to prepare for the exam. But in truth and in fact, you would need about adequately to comfortably finish this syllabus, 34 weeks, right? But you really have 26 teaching weeks, but you need at least 34 weeks or 34 solid classes to cover all 3 modules of the syllabus to be ready for your exam in mid-May to June next year. So how will you be assessed when you have your exam next year, God's willing? So there are 2 papers. There's paper 1, which you get an hour and 30 minutes to do 45 multiple choice questions, which is less. No, you're getting more time and you're getting less questions than you got in CXC. So CXC, you had 60 multiple choice, but now you're down to 45. So how it works is that you're given 15 questions from each module. So 15 from each module, and they're weighted equally. So CAPE is forcing you now to have equal coverage of the syllabus. So it doesn't even help you if you are excelling in one module and weak or didn't even complete the next module. If you didn't complete a module, guys, that is a serious infringement in CAPE, because everything is graded equally. Each module is graded and assessed equally. So your job as a student is to actually have complete coverage of a syllabus, all right? Paper 2 lasts for 2 hours and 30 minutes. It has 3 compulsory structured essay questions, 1 from each module. Each question is worth 30 marks. Again, each module in CAPE is graded and tested equally. So your best chance as a student is to be rounded or to be grounded in each module. You want to have complete syllabus coverage, right? You can't afford to get to the end and not do a module. Why am I stressing this? Well, not so much for biology, but more so for Unit 1 chemistry. The syllabus can be so demanding or so taxing that sometimes a module is not completed, and or sometimes because of the pressure, you may actually get one entire module as just classwork or as a project in some schools. So they'll assign different topics to different students, and students present to each other. and that's just to cover the syllabus and say that it was covered, but it doesn't mean that that was done sufficiently. But I want you to be aware of this so that whatever the situation that is going on at your school, you still know that it is your responsibility to have full syllabus coverage because you're going to be tested equally on each module. So paper 1 has 45 multiple choice questions, 15 from each module. All the questions are compulsory and knowledge of the entire unit is expected. The paper will assess a candidate's knowledge across the breadth of the unit, and it will be assigned. 45 questions will be weighted to 90 marks. You can be given questions using diagrams, data, graphs, prose, or other stimulus material. Paper 2, which is another 40% of your grade. So here's another difference now between CXC and CAPE. Now in CXC, the paper 2 had a greater weighting on your score, your final grade. In CAPE, it is equal. So the multiple choice is worth 40% of your final grade, and the paper 2 is worth 40%. What it means is that you cannot see the multiple choice as you saw it in CXC as this thing that you SWAT near the end and just look back for the answers and repeat. No, you have to be preparing meticulously for the multiple choice at the same time you're preparing for the paper 2 because they are weighted equally. All modules are weighted equally and the 2 papers are weighted equally. So that means that you have to be doing both of them. So this has 3 compulsory questions, 1 from module 1, 1 from 2, 1 from 3. Again, equal coverage of the syllabus. Knowledge of the entire unit is expected. The paper will be worth 90 marks, 30 marks per question, and will be attributed across the question subparts. Questions will be presented in structured essay format. The questions will test the skills of knowledge and comprehension, use of knowledge, and experimental skills. Answers are to be written in a question booklet. Let me just go back to the multiple choice. The multiple choice didn't say what skills they were testing. So here it is. Now that the labs, so you have labs as well to complete. And there are at least 48 marks that you're trying to get for each unit in labs. There are a total of 8 marks for observation and recording and reporting and recording, manipulation and measurement drawing, and then you have more weighting for analysis and interpretation and planning and designing. So you're trying to get the maximum you can get out of 48 marks from the labs. So let's look at this assessment grid for the CAPE Unit 1 Biology. So we have 3 modules. So we have 15 questions in paper 1 from each module, right? So that's 15 points, but which is weighted, it's doubled and weighted up to 30 points each. So there's a total of 90 points that you can get from paper 1. Paper 2, each question is worth 30 marks. That brings up to a total of 90 points as well, and those are raw marks. Paper 3, which is really your labs, it is that you're getting technically 16 marks from each module, which brings us up to the 48 points that we saw a while ago. And I believe this is if you did the alternative paper instead of doing the experiments. So essentially, the raw marks you're going for is 60 marks per module. All right, how many labs are you expected to do? Now, I was looking to see if they had indeed indicated the number of labs. Let me see if I can find it. I don't even think. Just give me a second. For some reason, it wasn't clearly stated as it was in CXC. So let me just double check. Okay. CAPE doesn't actually give a specific number like that. The syllabus actually says even a minimum of 2 labs is required. Oh, you're saying you were told 10? Well, the syllabus did not expressly state it. it says that the number of labs can vary between schools, but for them, the focus is really on the skills. That's what I'm seeing here from my research. It says you are allowed to vary the number of labs, but the focus is on the practical skills and the successful completion and assessment of at least 2 labs, they said. But the core requirement of CAPE Unit 1 Biology is demonstrating specific lab skills, not completing a set number of experiments. Students must perform and be assessed on at least 2 different lab activities to show competency in a particular skill. So if we are to go by this, and this was taken from the syllabus, if we are to go by that, then if it is that they have 1, 2, 3, 4, yeah, they have 1, 2, 3, 4, 5 skills, right? And you must be assessed on the skills at least twice, then it could mean that you'd have at least 10. I think that's where the 10 comes from then, what that you were told. The 10 per science would make sense because if you have to get at least 2 grades for observation recording, that means you'd have needed at least 2 labs. So 10 seems like the minimum, but it could have been less than 10 because you can actually do more than 1 skill in 1 lab. But at the end of the day, the percentage marks that you're going for is that you're going to get 40% of your final grade. Okay, so somebody said maybe the students are getting extra for practice. I'm going to talk about that some more. No, I don't, it's not that you're going to get extra marks for practice, but it actually helps you with your preparation in the exam. The more labs you do and the more you physically carry out the lab, is the more you're actually prepared for the lab-based questions, because there will be lab-based questions. But a lot of times, when you don't know a lot of labs, it means the other ones, you have to imagine it or you have to watch it on YouTube. Because if you never did it, you have to go and research it and look at how it's done. And you'll remember it less when you go and research it on YouTube than if you actually carried out the lab yourself. So teachers who can facilitate more labs is actually helping the student to actually become more intimately aware of the practicals, which will come in the papers, the exam papers. So yes, I was right here. So your multiple choice paper is going to give 40% of your grade. Paper 2 is going to give 40%. Your labs are going to give 20%. So of course, you're going to want to try to get into that exam with as much as possible of the 20% as you can get, right? You're going to want to get the majority you can get for the 20%, and of course, go and score the other 80. And as I said, the major thing which I'm kind of hoping you will walk away from this seminar with is that the 2 papers are weighted equally and that each module is weighted equally. If you remember nothing else and you remember that, then it's going to dictate your behavior, right? It's going to dictate your behavior because you're going to realize that, hey, I have to complete all these modules. I cannot give any preference to any one module, even though I like it, even though my teacher taught this longer, and I'm just doing a project for this one. It is my responsibility to learn it. And you cannot treat the multiple choice casually. The multiple choice questions are very hard. I wouldn't say hard that you cannot manage it, but they're not like CXC type multiple choice questions. So you'll have, like in CAPE chemistry, you'll have a multiple choice question, which requires like 6, 7 steps of calculation for a multiple choice question. CAPE will have multi-step questions as well. It's multiple choice, but for you to arrive at the answer, you'd have to go through multiple steps. So I don't want you to trivialize the multiple choice as you trivialized it, or some persons trivialized it in CXC. Question, do the multiple choice questions repeat? Definitely, yes, they do repeat, right? But what I try to do for the students who do classes with me or at our institution is that we try to expose you to the question bank, meaning that we look at the last 10 years of past papers, and we basically would look and see what type of questions they ask on a topic. So for example, say the first module when you start module 1 and you're looking at aspects of biochemistry. So you're finished with studying carbohydrates. Once you finish studying carbohydrates, what we'll do is look through 10 years and see all the multiple choice questions that came on carbohydrates. and then ask you to complete them as like a test, a quiz. So what we do at Extra Time is that we set up the multiple choice questions as quizzes. And once you go in, because we would have already selected the answers, so you would have gone in and you go through the quiz and you select, and it grades it automatically and tell you what you scored. And then we would discuss the questions afterwards, and you can review it and see what you needed to improve. So we find that for you to really be confident that you have seen the bank of questions, you have to look at about 10 years, not just looking at 1 or 2 past papers ahead of your year. So you're going to do your exam in 2026. You shouldn't be comfortable just doing 2025, 2024 and say, oh, I've done 2 past papers or 3 past papers. No. But if you have seen the bank for the last 10 years, they hardly have new questions, to be honest. So definitely you want to look at the past. You want to make sure you have been seeing the multiple choice questions because they will repeat. But the challenge with them, with the multiple choice, even though they're going to repeat, is that because the answers are so complex, right? If you don't actually remember why the answer is what it is, it's not easy to just swat it like that. So you still have to understand why. And once you have done it before and you understand why, you'll remember. So yes, thankfully they do repeat them. So in order to fully be prepared, like by getting through all 3 modules sufficiently, and I mean to the sufficient depth that you need to get to, you have to dedicate no less than 3 hours to the preparation for each week, right? And in CAPE, in CXC, it was very important, but in CAPE, it is a must. You must do past papers right after you've learned a topic. So how we structure our program here at Extra Time is that once you learn a topic, so once you have a class, and our classes start next week, Tuesday, once you come, whatever you learn, we would have looked for past papers that tested you on that section that you learned. So we won't give you an entire paper or an entire question. We'll just find a portion of the question that you can manage based on what you did in that class. Not even looking at what you did in the class before. All right, so we want to test you only on what you just covered. And we want you to do the past papers as soon as you learn the topic, because you're automatically simulating the exam. You're automatically applying the topic, the knowledge, sorry, and it will be retained longer in your head. And the greatest thing about doing the past paper right away is that you immediately see the link between the theory and the question. And then it makes you see that, maybe something that I was glossing over, look at this, they're asking me in a past paper. So then you make a note and you're like, no, this is something that's important. I need to study this because see them come and ask me. So what you find, guys, is that when you are studying or when you're learning things at this advanced level, as I said, CXC was important, but no, you can't do without doing that. When you're learning things at an advanced level, because you're given a large volume of information, you sometimes don't know what part of the information is more significant than the other. And so you do not know really how to do focused study. But once you look at the past papers that are related to that topic, immediately it gives you clarity and it teaches you on what aspects you must focus. So your studying becomes way more efficient when you glance at the past papers. So that's one of the reasons why we do it every week. And secondly, as I said, because the multiple choice and the essay have the same weighting, then we have to do both types of questions not every week for the multiple choice, but as soon as you've finished a topic, we find the multiple choice questions, but every week you do essay type questions. Also, in a way that we do it at Extra Time, the fact that we allow you to submit it and mark it before we discuss it, because we could have just given you the answers, we could have actually asked, look for the questions and then giving you the answers. But that extra step of having you attempt it and you submitting it, and we mark it and grade it according to the CAPE mark scheme and give it back to you, it allows you to understand how competent you are in scoring marks for CAPE, because the CAPE mark scheme is way more meticulous is way more meticulous than CXE. So you might find a question in CAPE worth 5 marks, right? And you might think, okay, that's 5 points. 5 marks, 5 points, because that would be how you'd have probably thought it would be graded in CXE. Lo and behold, 5 marks in CAPE could mean 10 points, and they're giving 0.5 mark per point. No, it's only with practice where you are continuously submitting work and we are using the mark scheme and marketing. And when you get it and you think you submitted 5 points and you look at it, you get 2.5 out of 5. That's when you're going to pull up your socks. And you're going to be like, what else did I need to do? And then you are going to be seeing, you're going to see the answer. So they're going to know, oh my, I should have added more of this, should have done that. Another thing with CAPE as well, why it's important to do the past papers and have them marked, is where they're not going to come out directly a lot of times and say to you, oh, you need to draw a diagram. They're not going to say it. They're going to ask you something like, for example, in genetic engineering, like we saw in the video earlier about cystic fibrosis and how genetic engineering could cure it. And let me ask you something like, say how genetic engineering helps with cystic fibrosis. Now, your natural tendency from CXC is that you start writing, and you start writing, and you start writing for 8 marks, only to realize that a good 4 marks out of that 8 mark was allocated for a diagram. They never asked you directly for a diagram. But in CAPE, you are expected to make those assumptions and draw a diagram. Anything that actually has a diagram, draw it. And they have allocated marks to it and they never asked you directly for it. And so you might have been writing away and never ever get the 8 marks. But these little things will only come up when you actually do the questions and actually have the questions reviewed and marked. Then it will come back to you and say, oh my God, I needed a diagram here. And you, hopefully a wise student would go and look for the diagram. And so the next time, as I said, we are making sure you're practicing the past papers every week. So by the time we get to term 3 now, or the last part now where you are now preparing directly to sit the paper and we do like an examination marathon or something, this is now the second time around that you're seeing past papers. And so you will remember the previous encounter that, oh, things like this requires a diagram, And so it makes you way more targeted and way more efficient in answering your questions. And CAPE is very big on presentation. So they don't want you to just be writing paragraphs. You need to present bullet points, tables, diagrams, all graphs, if you can use graphs to illustrate your answer. So it's really forcing you to grow to a higher level in your academic studies, CAPE. So they give you less topics, but they give you depth, and then they're also examining you to a greater degree, because now they're expecting that you would have matured as a student. So let's see now the grading. All right. So CAPE has, of course, the 3 modules that you got. The profile reflects the 3 modules. So for example, the student got a 1 in Unit 1 Biology because the student was competent in all 3 modules, Module 1, 2, and 3, scoring A's in all 3 modules. Now, CAPE goes all the way up to 7, guys. This is not so clear. Sorry about that. But up to grade 7. Let me see if I had, unfortunately, I'm not so sure why it's so blurred, but it goes up to grade 7 and up to grade 5 is a pass. So if you get a 5, they say that it's acceptable. Grade 4, satisfactory. Grade 3, they said you have a good grasp. Grade 2 says you have a very good grasp. And if you get a grade 1, it says you have an exceptional grasp. And our aim is to help you to get that grade 1, our exceptional grasp. But a grade 2 is very good, right? But really and truly, this is where we want you to stay and keep. So I suggest that you all have your personal copy of the syllabus, and you need to tick off the objectives as they are covered every week. You have to be alert and aware as you're studying, right? Yes, your teachers will be looking at the syllabus as well, and will be preparing you from the syllabus. But when you are following along, you become a more aware student, right? And so, and you become more targeted in your studies. And what it means sometimes is that there might be an objective and you are taught, but you don't feel comfortable with it. And you can seek alternative sources of information to cover that objective, and then you tick it off and say, yes, I've covered this satisfactorily. This is an excerpt from the syllabus. So CAPE will give you specific objectives. For example, explain the process of diffusion, facilitated diffusion, osmosis, active transport. These are all of these. It goes on, it explains what are some specific things they want you to know. Like they want you to be able to distinguish between diffusion and osmosis, passive and active processes. Diagrams are required. No calculations will be set on water potential. So this tells us as a teacher how deep we should go. But then they also have suggested practical activity. This is a topic that can have a lab on it. And so a suggested practical activity is that you investigate the effect on plant cell immersion into solutions of different water potential. For example, you immerse onion epidermal cells in different solutions of different concentration. Now, your teacher at school may get to do this suggested practical activity or may not get to do it. But this is a trick. The fact that they have listed it as a suggested practical activity, it means that you still have to be aware of it. So what we do is that we try to include it in the topic itself, in the subject itself, include this lab when we are teaching the subject so that it's a part of your body of knowledge. But just in case, if you are following along and you notice that, oh my, you didn't do a lab on this and your teacher didn't mention anything, you know that it's your job to still go on YouTube and research this and watch it and make sure you see how it's done, because this is something that they can still include in the exam as a suggested practical activity. So read ahead of your experiments. Once your teacher tells you what experiment you're going to do, you need to read ahead of it. Meaning, sometimes I know it's difficult in CAPE because a lot of times you might be doing module 1, but the next thing you know, the lab is on a module 3 topic. I've seen that a lot of times. 100% I've seen that. And because the teacher is just trying to get the labs done in the time that they get to do it. So it's not a malicious thing, but you end up getting a lab on a topic you haven't learned yet, and you need knowledge to be able to interpret the lab. Well, luckily for you guys, with the technology, ChatGPT and YouTube, you can quickly arm yourself with enough knowledge to understand that lab so that you can be able to write a proper discussion that sufficiently analyzes the points. And watch YouTube to see how the lab is done before you go into the lab to do it, right? So, like, for example, the suggested practical that we saw a while ago that CXC, that CAPE had suggested, you could have gone on YouTube and looked it up and says, what happens to epidermal cells? And a lab would be there. You could have seen it. So if you never got to do it in your own school, you would have still seen the experiment. And so you'd be prepared for a question on these epidermal cells because you researched it. So those are some of the things I just want to make sure you're aware of as a student starting to do CAPE Unit 1 biology, that yes, it goes down to a grade 5, but most schools don't really consider it to be serious if you're not getting a grade 1 or a grade 2 in CAPE. Grade 3, maybe, but beyond a grade 3, they still, even though technically it is stated to be a pass, most persons will still look at something beyond a grade 3 as not satisfactory. So I don't want you to get into that comfort zone. So I've still passed. And of course, you'll pass. If you're just doing the subject to say, all right, I need 2 CAPE subjects, and I do have 2 CAPE subjects, no problem. But if you're looking at programs which are competitive, any program at university level, you're not going to want to have less than a 1 or a 2, because then that's going to, like some areas like medicine, that's going to affect whether or not you get a scholarship or whether or not you get government subsidy. And that's gonna cost you money in the long run, versus if you had invested the time and effort now and got grade 1, right? So those are some of the things I just want to leave with you. Do you have any questions though, what we've covered so far? Any questions? All right, guys. So thank you so much for tuning in. The chemistry starts shortly in the next 10 minutes. So I'm going to end this, and in the next 10 minutes, we will log in back for the chemistry if there are no questions.
I had posted one in the chat.
Oh, yes,
new
I did
to this
respond.
platform.
Could you repeat that?
No, I was saying that I'm new to this platform, so
Oh,
new
yes,
to
I saw your
exercise.
question and I had sent a response.
Okay.
Yeah,
All right.
we'll on WhatsApp.
All right. Thank you.
All right, guys. All right, so enjoy your day and see the rest of you shortly.